In this video, we are going to learn about the microscopy in detail. First of all, fluorescent microscope. A fluorescence microscope is much the same as a conventional light microscope with added features to enhance its capabilities. The conventional microscope uses visible light nanometers, about 400 to 700 nanometers, to illuminate and produce a magnified image of a sample. A fluorescence microscope, on the other hand, uses a much higher intensity light source, which exhibits a fluorescent species in a sample of interest. This fluorescent species in turn emits a lower energy light of a longer wavelength that produces the magnified image instead of the original light source. This fluorescent species in turn emits a lower energy light of a longer wavelength that produces the magnified image instead of the original light source. Fluorescent microscopy is often used to image specific features of small specimens such as microbes. It is also used to visually enhance 3D features at small scale. It is also used to visually enhance 3D features at small scales. This can be accomplished by attaching fluorescent tags to antibodies that in turn attach to targeted features, or by staining in a less specific manner. And the reflected light and background fluorescence is filtered in this type of microscopy. The targeted parts of a given sample can be imagined. This gives an investigator the ability to visualize desired organelles or unique surface features of a sample of interest. Confocal fluorescent microscopy is more often used to attenuate the 3D nature of the sample. It is achieved by using powerful light sources such as lasers that can be focused to a pinpoint. This focusing is done repeatedly throughout one level of a specimen after another. So, what is confocal fluorescent microscopy? It is often used to attenuate the 3D nature of samples. This is achieved by using powerful light sources such as lasers that can be focused to a pinpoint. This focusing is done repeatedly throughout one level of specimen after another. Most often, and large image reconstruction program pieces the multi-level image data together into a 3D reconstruction of the targeted sample. So when we just look at the structure of this particular fluorescent microscopy, first there will be a specimen on the bottom and object will be there, and in the mirror will be there excitation filter, detector and emission filter. These are the parts. In most cases, the sample of interest is labeled with the fluorescent substance known as fluorophore and then illuminated through the lens with the higher energy source. The illumination light is absorbed by the fluorophores now attached to the sample and causes them to emit a lower energy wavelength light. This fluorescent light can be separated from the surrounding radiation with filters designed for this specific wavelength, allowing the viewer to see only which is fluorescing. The basic task of fluorescence microscope is to let excitation light radiate the specimen and then sort out the much weaker emitted light from the image. First, the microscope has a filter that only lets through radiation with a specific wavelength that matches your fluorescing material. The radiation collides with the atoms in your specimen and electrons are excited to a higher energy level. When they relax to a lower level, they emit light. To become detectable or visible to human eye, the fluorescence emitted from the sample is separated from the much brighter excitation light in a second filter. When they relax to lower level, they emit light. To become detectable or visible to human eye, the fluorescence emitted from the sample is separated from the much brighter excitation light in a second filter. This works because the emitted light is of lower energy and has a longer wavelength than the light that is used for illumination. Most of the fluorescence microscopes used in biology today are epifluorescence microscopes, measuring that both the excitation and the observation of the fluorescence occur above the sample. Most use xenon or mercury arc discharge lamp for the more intense light source. The refinement of epifluorescent microscopes and advent of more powerful focusing light sources such as lasers has led to more technically advanced scopes such as the confocal laser scanning microscopes and total internal reflection fluorescence microscopy, which are the two types. Confocal laser scanning microscopes. Confocal type. Confocal laser scanning microscopes and internal reflection fluorescence microscope. Confocal laser scanning microscopes are invaluable tools for producing high resolution 3D images of subsurfaces in specimens such as microbes. Their advantage is that they are able to produce sharp images of thick samples at various depths by taking images point by point and reconstructing them with the computer rather than viewing whole image through the eyepiece. These microscopes are often used to do imaging structural components of small specimens such as cells, conducting viability studies on cell populations. Are they alive or dead? Imaging the genetic material within the cell, viewing specific cells within a large population with techniques such as fish fluorescent in situ hybridization. Next one is the interference microscopes. Although all optical microscopes in the strict sense Although all optical microscopes in the strict sense create images by diffraction, interference microscopes create images using the difference between an interfering beam unmodified by the specimen and an otherwise identical beam that illuminates it. A beam splitter divides light into two parts. The beam splitter divides light into two parts, one of which passes through a specimen while the other bypasses it. Then the two beams are combined. The resulting interference between them reveals the structure of the specimen. The first successful system invented by British microscopists Francis Smith and Francis Franson in 1947 used quartz lenses to produce interference and image forming beams that were perpendicularly polarized. Although this worked well for continuous specimen, in the case of particulars it was better to have the reference beam pass through a bare area of the specimen preparation, and by 1940 the use of half silvered surfaces and highly tapering slides allowed polarized light to be dispersed with. Meanwhile, different interference contrast has developed by Polish born French scientists. A beam splitting Wollaston prism emits two beams of polarized light that are plane polarized at right angles to each other and that slightly diverge. The rays are focused in the back plane of the objective. Then they pass through a composite prism that is isotropic at the midpoint with an increasing optical path difference away from the midpoint. The background color of the image depends on the setting of the prism, which can be slide longitudinally to produce a spectrum of colors that vary through the spectrum to black. The sensitivity is greatest in the middle position, but the color contrast is greater when the strong background tint is selected. Most recent developments include asymmetrical illumination contrast and modulation contrast, which exploit offset of oblique illumination. Now let's learn about the parts and other information about interference microscopy. Over the past few years, the microscopic techniques had faced with some complications with the observation of living specimens as transparency, light, and because of that doesn't have significant contrast. Thus, with the development of these techniques, it's possible to visualize specimens with adequate contrast. Interference microscopy or quantitative interference microscopy. Interference microscopy or quantitative interference microscopy is one of these techniques that derive from phase contrast microscopy but is more sensitive than this technique and make possible the easy and clarity viewing of living organisms. This technique is used by taking light from a condenser and using a prism to separate the light into two beams. Then one beam, object beam, goes through the specimen and the objective, and the other beam or reference beam goes through the another objective without touching the specimen. These beams allow a specimen to see through the difference in fields caused by the two beams, and the differences of the two images allow details to be seen. Differential microscope contrast microscopy. There is a variation of interference microscopy called differential interference contrast microscopy, also known as Nomarsky interference contrast microscopy or simply Nomarsky microscopy. This optical microscopy illuminates techniques used to enhance the contrast in unstained or transparent samples was named after this inventor and also uses two beams produced by a single polarized light. Initially, the polarized light is divided into two rays polarized to each other, sampling and reference rays, which enters in the first Nomarsky modified prism, Wollaston prism. Then these two rays are focused on the condenser for passage through the sample and travel to adjacent areas of the sample, divided by the separation is normally similar to the resolution of the microscope. After that, they will face different optical wavelengths where areas differ in refraction index or thickness, which will cause a change in phase of one ray relative to the other according to the delay experienced by the wave in the more optically dense material. Lastly, the rays go through the objective lens and are focused to the second Nomarsky modified Wollaston prism, which joins the two rays into one polarized, which make an image with a three-dimensional appearance. This final junction of rays leads to interference, brightening or darkening the image at that point according to the optical wave difference. This interference technique have advantages in uses involving living or unstained biological samples, especially their application in biology, crystallography, mineralogy, and chemistry. In standard optical microscopy techniques, its resolution and clarity is also visible. On the other hand, these techniques also have limitations in its requirement for a transparent sample of similar refractive index. Differential contrast microscopy is inadequate for thicker samples, tissue slices, pigmented cells, and for most non-biological samples because of its polarization dependence. Next technique is the electron microscopy. It is a fantastic tool that enables biologists to capture images of their samples at a greater resolution than with light microscope. There are several types of EM, and each of these can provide different information about your sample. The large field of EMs expanding at the time. There are many advanced instruments that I do not describe below. In this article, I will discuss the basis, the main types of EM, that information you can obtain, and the challenges faced by biologists. First is the magnification and resolution. Often the term magnification is used when discussing the power of a microscope. However, magnification is not the main issue affecting microscopes, it is the resolution. Resolution is the ability to distinguish two objects as separate. Imagine a car coming towards you at right. Initially, you would see a single headlight, and at the same point, you would be able to separate the light into two distinct headlights. This is the minimum resolvable distance or resolution. In microscopy, we refer to the diffraction pattern created by imaging a point object. The resolution of two objects in a microscope depends on them being sufficiently separate so that the diffraction patterns do not merge, known as the raying criterion. Ernest Abbe was able to determine the optimum resolution that a microscope can achieve, and the equation for this is the Abbe diffraction limit. Microscopes are designed to minimize variables so that the main limiting factor is the wavelength used to image the sample. All electromagnetic radiation, light, X rays, radio waves, etc., has a set wavelength. Using a form of electromagnetic radiation with a smaller wavelength increases the resolution that can be achieved. Light microscopes can achieve a resolution of 200 nm. Super resolution light microscopy allows some biologists to go beyond this limit, and a few X-ray microscopes are now also available. However, EM remains the main technique used by biologists for high resolution imaging of molecules, viruses, and cells. Electrons are negatively charged subatomic particles that have a wavelength. The wavelength of an electron is determined by using de Broglie wavelength and is linked to an acceleration voltage used to form the electron beam. For example, an AV of accelerating voltage of 100,000 volts resulting in an electron wavelength of 0.00337 nm and a resolution that is up to 1,100 times smaller that can be achieved with light. While it is possible to achieve subatomic imaging with an electron microscope, it is not possible to image biological samples at this resolution. As we can see, the biological issue is always the limiting factor. Next, instrumentation. There are several different types of EM, which can be split into two main categories like transmission electron microscope and scanning electron microscope. The main difference between these are in the optics, how the signal is detected, and the type of information you can obtain. So when we learn about both types of the CM, have an electron gun which contain an electron source, a filament that produces a cloud of electrons, a Wehnelt cylinder to form the beam, and an anode to accelerate the beam. There are three main types of electron sources: a tungsten filament, a lanthanum hexaboride crystal, and a field emission filament. First, let's learn about the transmission electron microscopy. The TEM transmits the beam of electrons to the thin sample onto a screen on a camera or detector. It has a large number of lenses. The condenser lenses, depending on the microscope, are responsible for the amount of illumination that reaches the sample and control beam intensity or brightness. The objective lens focuses the beam of electrons into the sample and applies a small amount of magnification. The intermediate and projector lenses magnify the beam and project it into the camera on screen to perform an image. It takes only a few seconds to obtain a micrograph or microscope image. The image is a result of the projected beam intensity. Transmitted electrons are detected as light areas in the micrograph. Darker areas occur where electrons have been scattered or absorbed by the sample. It takes only a few seconds to obtain a micrograph or microscope image. The image is a result of the projected beam intensity. Transmitted electrons are detected as light areas in the micrograph. Darker areas occur where electrons have been scattered or absorbed by the sample, thus reducing the number of electrons reaching the camera or screen. This is known as the bright field imaging, and this is the most common type of imaging for biological samples. The TEMs are often classified based on the accelerating voltage they are capable of. Routine TEM, that is transmission electron microscope, for biological imaging should be capable of an accelerating voltage up to 120 kilovolt. Most thin section TEM will be conducted using 82,000 kV. Advanced TEM techniques may require instruments capable of AE between 200 kV and 3 MV, which represent a resolution of 100,000 to 3 million times smaller than light microscope resolution. Next is the scanning electron microscopy. The scanning electron microscope focuses the beam of electrons into a small spot that scans across the surface of a sample. The condenser lens assembles the electrons into a fine beam. The objective lens focuses the beam onto the sample. Deflection coils cause the beam to move in a rectangular X and Y direction, producing a raster scan across the surface of the sample. The signal is transmitted to a computer screen. Reducing the area being scanned results in an increase in magnification, and some images formed from signals that are emitted from the sample as a result of a specimen beam interaction. Most biological samples will generate images using two types of electrons. Secondary electrons are low energy electrons produced by small energy transfer between the electrons from the beam. and electrons orbiting atoms in the sample. The energy transfer causes the orbiting electrons to leave the atom and become a secondary electron. An outer orbiting electron will then release some energy in order to jump into a gap left by the secondary electron. The second type, backscattered electrons, are high energy electrons that have passed close to the atomic nucleus and been reflected back or backscattered out of the specimen. In addition, there are a few applications that require the detection of characteristic X-rays, energy dispersive X-ray spectroscopy, or photons like cathode luminescence. There are different types of detectors to detect and collect the signals. Secondary detectors are low energy electrons, and only those produced near the surface can be emitted. The signal detectors are now not cameras, and the resolution of the same image depends on the spot size of the beam as it hits the sample and the interaction volume between the beam and specimen. The interaction volume directly relates to the AV of the beam. Biological SEM typically uses an AV of 1.5 kV for the best resolution. The SEM image is inverted compared to the TEM. Bright areas of the image are the result of more electrons being scattered from topography of heavy element staining. Relative large biological samples can be imaged using an SEM, as we no longer have to transmit the signal through the specimen. Now let's learn what are the challenges with biological EM. There are several challenges for researchers working on biological applications of EM. Vacuum. The electron beam operates with a fairly high vacuum. This causes problems for biological tissue as evaporating water destroys structures being imaged. This also means that it is not possible to image living tissues with electron microscope. Several protocols stabilize the sample during preparation. Samples can be frozen rapidly and imaged in their hydrated state in a technique called cryo-TEM, or they can be fixed with chemicals, dehydrated in solvents, strained, embedded in resin, and sectioned. Next is the lack of contrast. Biological tissue does not diffract many electrons. Electron dense stains or advanced EM techniques are often necessary to visualize biological ultrastructure. Next, transparency. The electron beam must be able to get through the sample in time. Some samples are small enough that they can be imagined whole, such as viruses, but most cells and tissues need to be sectioned. Biological samples are non-conductive, which can create issues when bombarded with a negatively charged electron beam. The samples can be unstable and drift, blurring the image or create distortions in the signal, all of which are features of an accumulation of negative charge or charging. Heavy metal staining will help prevent this, and a conductive coating is applied to the sample to dissipate the charge.